Hello, hello and welcome to the GECCO conference. I'm going to be presenting on a simple evolutionary algorithm guided by local mutation for an efficient RNA design. So I will start by giving you some biological context. What are RNAs and what are the main roles that RNA play in our body, in our cells, more specifically? So RNA stand for ribonucleic acid, and compared to DNA, they are quite very similar, but with two main differences. RNAs contain ribose and they are pentose as their pentose sugar instead of deoxyribose for DNAs. And also RNAs contain uracil as their pyrimidine instead of thymine. So DNAs generally get more attention, but RNAs play also very important role in all our cellular processes. They can be generally grouped in two main categories, non-coding RNAs and coding RNAs. Our main focus here would be non-coding RNAs, even though coding RNAs also play roles, but those are RNA that are translated into protein, so they generally play only information storage role. So one of the main biological process in which three RNAs play very important roles is protein synthesis. So protein synthesis happens in two main phases or steps. The first step is the transcription, in which only coding RNAs play a role where they store information needed for the translation into protein. And then during the translation, there are two main non-coding RNAs called tRNA or transfer RNA and ribosomal RNAs that play important role in this and that stage of the translation. So for non-coding RNAs to perform biological function, they need to fall into a three-dimensional structure, which is displayed here. So, and in general, the three-dimensional structure strongly depends on the secondary structure, but naturally, it is basically taught that RNAs are folded in the very hierarchical way. So where we first have a secondary structure and then subsequently the secondary structure is refined into a tertiary structure. There are generally two main ways to perform this operation, either experimentally or computationally. And computational methods are generally based on two main models, either machine learning or statistical methods or thermodynamic methods. In this way, we'll be using RNA-fold as the folding tools, and this tool is based on thermodynamic model. So unlike RNA folding, RNA design itself is the reverse process in which the biological function is known and the goal is to figure out what are the RNA sequences associated to that biological function. But if we code it just on secondary structure, if we suppose that only secondary structure is known, and then we would like to find RNA sequences that fall into that secondary structure, the problem itself would be called the inverse RNA folding problem. So we can understand it as an optimization problem in which a given target is sigma star, and then we know the length. And then now the goal will be to search in the space of all possible sequences of this fixed length, a subset of sequences that fall into that target structure. And the picture here displays clearly a successful case for an RNA inverse folding tools. So many RNA inverse folding tools have been proposed in the literature, from the oldest one, which is RNA inverse, to the newest one, RNAs, that perform more sophisticated methods like machine learning or anti-RNA that perform some ant colony optimizations. Our main goal here will be to try to provide an improvement to the existing tools, understand the evolutionary algorithm setting, and find a better way to explore the RNA search space quickly. So how does an evolutionary approach apply to the inverse folding problem? Here is the general principle. For a given target structure, we start by initializing a population of RNA sequences compatible to that target structure. And then we would perform genetic operation on that population until a subset of sequences are found to fall into that target structure. In this work, we will be focusing our evolutionary algorithm more on selection and mutation genetic operations. We are not the first, of course, to think about applying evolutionary algorithm on this problem. Moderna is the first tool developed by Antico that performs a multi-optimization scheme. And then 4 years after followed evolutionary RNA design proposed by Ali and his collaborators that uses more sophisticated techniques where the target structure is decomposed into subcompartments, and individually those subcompartments are being optimized using evolutionary algorithms. So here we propose a novel framework which is conceptually very simple, but relying on three main objective functions I display here. The first one helps to measure the similarities to the target structure. The second one, the stability. And the third one, combine both. So one of the main core of our algorithm that we propose here is the mutation operator. Here we propose a simple mutation operator that rely on two main parameters: the distribution of, or the probability distribution of the nucleotide, and the probability distribution of canonical base pair or possible base pairs for aRNA sequences to form. So here is an example illustrated in the picture, where in the middle we have a sequence that the goal would be to mutate. And then we first start by selecting a random position that will be mutated, and then pick now a nucleotide that will replace this random unpaired position based on the probability distribution Pn. And then the same thing can be also done for the base pair position, but picking a canonical base pair and mutating simultaneously the two corresponding base pair positions. So how does those three functions come together to actually play a role in our evolutionary framework? We start by initializing a mixed compatible RNA and unfolded RNA sequences. And then we first evaluate them based on the first objective function. And then we keep then the test best sequences based on this objective function. And then we select n RNA sequences from the population based on now the second objective function. At the fourth step, we then mutate those sequences to generate new RNA sequences. And then we can then repeatedly perform the second, the third, and the first step until a number of sequences are found to fall into the target structure. So after finding those subsets, we can then start refining those sequences by just changing the second objective function, replacing the second objective function in the same evolutionary setting by the ensemble defect one, so the third early mentioned. So to assess the performance for our tools, we use two benchmark data sets. So a non-eternal benchmark data set and an eternal data set. So it's a set of 63, the first one, more experimental target RNA secondary structure collected mainly from R-farm, and the second one is collected from eternal genes online. So for the following tools, we use here two energy parameters. So the tuner energy parameters proposed in 1999, which is the oldest one. And we use it only to benchmark the eternal data set. And the second energy parameters is used to benchmark both for the sake of robustness on the energy parameter change. So here is the first result that we obtained. Our tool is aRNAque, it's called aRNAque, and we implemented it in Python. And the table here shows that our tools solve much more target than most of the previous or existing tools, but only 2 or 3 targets less than RNA and ERD tools, which places our tools third on the list. The results from other tools were taken from same RNA paper. So the second benchmark was performed on eternal, and then we used first the second parameters and secondly the ordered parameters, energy parameters. So for the newest energy parameters, aRNAque shows to be on top of the list as displayed in this table by solving 72% of eternal dataset. Here we benchmark only 4 tools, NEMO, ERD, Learn RNA, and RNA Inverse, because in the earlier paper and benchmark data, they did not actually benchmark them on the newest energy parameters. But compared to the oldest benchmark on the old energy parameters, aRNAque seems to be placed just behind NEMO by solving 3 structures less than NEMO. So we also address the quality or compare the quality of the sequences designed with the one generated by NumPack, because NumPack is the only tools here actually minimizing the third objective function, which is the ensemble defect. And both the minimum free energy and the ensemble defect seems to be well minimized using aRNAque as well, and even better minimized. And the Hamming distance distribution clearly shows that aRNAque outperformed the design sequences produced by NumPack. So the same comparison was drawn on the 3 benchmark tools that we used on the using the newest energy parameters. And then down there we can see the Hamming distribution of the sequences design, and we see that the median of most of the aRNAque sequences seems to be 0, which means the aRNAque mostly find some sequences that fall into the target. And for the energy and the ensemble defect, aRNAque seem also to produce qualitatively good sequences. We further compare the computational time of aRNAque with RNA Inverse, which was the second on the list. So in the inset plot, we can see that as the CPU time of RNA Inverse grows exponentially with respect to the length of the target, the aRNAque CPU time does not. And instead of the quality or the number of sequences found, aRNAque dominated by 7 as displayed in the previous table. And we can see there downstairs the plus that shows the sequences that aRNAque were able to design in the short amount of time compared to the average time needed by RNA Inverse, and less sequences were designed by RNA Inverse that were not designed by aRNAque. So as a conclusion of this work, we can say we propose a very simple, conceptually, evolutionary framework that actually help us to fit explicitly some sequence constraint, and it shows fast computational time and qualitatively good design sequences. It is also robust on existing energy parameters. As a perspective of this work, we would like to extend our algorithm to pseudonodes, improve the diversity of sequences since we are using only local mutations, and then we would also like to test experimentally the design sequences. Thank you for your attention.